Hello viewers, I'm going to give you a lecture on laboratory course of life sustaining system of animal physiology. My today's topic is smear preparation and identification of human blood cell. Uh, it is very questionable why human blood has been selected for identification. I like to mention you some peculiarities of the human blood. It has two types of cell, nucleated or non-nucleated. It has a peculiar substance that is platelets, that is thrombocytes, which is very specific in human blood. Why it is specific? Because it helps in the blood clotting system of the human blood. The cells are not only nucleated, non-nucleated, it is also of different forms. That is, it is heteromorphic in character. It functions differently, though it is heteromorphic. Again, the cells are medically important very much today because counting of the TC, that is the total count of the blood cells, hemoglobin, erythrocytes, and the leukocytes, differential counts of the leukocytes, ESFL, platelet counts, everything is usable in case of the determination of pathogenicity of human body. Not over that, moreover, human blood is used in homeostatic strategy. It does the various types of functions such as respiration, transportation of oxygen particles, excretion, and by doing all other every functions, it keeps the homeostasis of the body. Well, so we have got interest to identify the particular cells. So I want my discussion is step by step. I want to know first the what is the chemical characters of those cells which can react with my staining capacity. Number one. Number two, I want to visualize the all cells for identification. Thirdly, the anatomical variations will be determined to and observed for proper identification of those cells. Now, in the first point, that is the chemical nature of the blood, I have to find out. The blood cells in the human body. The nature of the what is the nature of the blood cell? It is a connective tissue. The connective tissue is mobile. That means it is a fluidity determines it is a fluid connective tissue. Its fluidity is maintained by blood plasma, which remains in the system at 55% ratio, in which the other component, that is the blood corpuscles, remains there at 45% ratio. This 45% ratio is the packed cell volume. That means without blood plasma. The total volume is about 5 to 6 liter. Otherwise, I can say that it is the 10% of the human body weight. Anyway, my concern is regarding the blood corpuscles. The blood corpuscles are heteromorphic, already I have told. The heteromorphic character has been determined. That means the classification of these corpuscles has been determined mainly on the three factors. One, the nucleus, second, the corpuscles, and third, the most important, the chemical reactions of the cytoplasm. Nucleus may be present or may not. It is not present in RBC and platelets, but present in WBC. Another character, the granules, it also may present or may not. It present in the neutrophil, eosinophil, basophil, but does not have in monocyte and lymph. So granules are there in different forms, and so that it is identified as different way. So identification of the granules are most important because to know their functions separately. But peculiarity is that all the corpuscles do not behave in the same manner because of their different pH factor. Some of the particles, that means some of the granules, are acidic in nature, some are basic in nature. So we require now to stain these particles. When both the characters are there, so we want such type of stain which can stain the acidic particles, simultaneously stain the basic particles. When you choose the basic stain, for example, methylene blue, it stains the basophilic substances, which is acidic in nature. For example, nucleus of the cell, endoplasmic reticulum, DNA, RNA. When we choose the acidic stain, for example, eosin, it has the affinity for the acidophilic or eosinophilic substances that are actually basic in the nature. For example, cytoplasm or granules. But we have to stain the blood. So such type of staining material we have to choose that can help the staining capacity of two types of corpuscles simultaneously. Yes, we have chosen this staining material as leishman stain. Blood smears is stained with it because it is a neutral and differential stain. Why it is differential? Because it contains both type of staining material, eosin and methylene blue, that is acidic and basic, both type of staining material. Eosin and methylene blue, if we consider their configuration, we can see this structure of methylene blue, that is amino group, here, and here, is responsible for its basic character. And as like that, eosin blue, this character, that is COO, and the hydroxyl portion is responsible for its acidic nature. Peculiarity is that due to its different characteristics, when this stains a cell, it proceeds their staining property in differential diffusion rate. Due to its diffusion rate, they migrate to the staining property to the cellular component in different times. So we require to maintain the time of staining span in proper way. The staining time will be discussed later on. Now we have to prepare the reagent. Commercially, it is obtained from the market as leishman powder. That leishman powder is taken at 0.15 gram and it is mixed with the methyl alcohol 100 ml in quantity. The mixture is heated at 50 degrees centigrade for 15 minutes. Then it is filtered and kept at 37 degree at room temperature in the laboratory. But it is better to prepare it 2 hours before the experiment. This leishman powder has two components you have noticed. One thing, the powder with two types of material, staining material, and dissolved in methyl alcohol. It is to remember that methyl alcohol is added there as a fixative of the cell. Number one. But attention to do that methyl alcohol should not contain any acetone, because acetone will destroy the cell structure, cell wall, and the total cell will be distorted, and staining will be barred by this particular chemical, that is acetone. So methyl alcohol should be acetone free. Now staining material is ready for the reaction. Hello. Now the process of visualization and identification of the species, that is the identification of the blood smear, should be discussed now. Leishman, other than the leishman stain, we have taken another sample, that is the blood, human blood as sample, pre-slide, grease-free, 2 disposable hypodermic syringe, distilled water of different pH medium, absolute alcohol and cotton, microscope of 2 objective size, that is 10x and 45x, a dropper pipette. This requirement is enough to make the experiment. Now the process of smear. I'm going to smear the blood cell. You have to remind it. During this function, we are making the smear on the slide. So first you have to clean the slides. Cleaning means any dirty particles should not be there. The cleaning will be done with the alcohol and water. Now it should be dried. Why? Because if it is not dried, then the heat fixation, that is the dry fixation of the cellular particles or the cellular corpuscles, will not properly adhere to the surface of the glass slide. Now the second stage. In the second stage, our function is to sterilization of the material, sterilization of the structure from where I am collecting the blood, that means our fingertip. Fingertip will be sterilized by cotton swab with alcohol, absolute alcohol, and needle will be steriled by burning the tip over the blue flame of the burn. Now, after the pricking, blood has been oozed out. The blood will be collected at the one end of the slide, right? Now the spreading of that blood. One drop of, if you notice the drop of the blood or droplet of the blood, then you will see the over the blood there is a rounded structure due to surface tension. You should touch the second slide just over that surface tension of the blood, so that by means of the capillary action the blood spreads sidewise horizontally. Then touch the first slide. Now third function is to push the slide, that means the second slide, at the same equal speed towards the forwardly way. Now this function will be possible only when you keep the slide or hold the second slide at 45-degree angle. Otherwise, the smear will be thick, and if it is thick, the corpuscles will not be distributed evenly on the slide for future verifications. Now smear should be dried. Smear, the peculiarity of the smear, the vision of the smear will be conical. Vision of the smear will be tongue separate, and the smear of the color of the smear will be reddish, light reddish. This smear is dried then in the air for few minutes, 1 minute or 30 seconds. Now smear is ready. Now come to the preparation of staining. Yes, it is the process, visualization, for your visualization, how you should make the slide smear. Okay, this is the blood droplet. This is the attachment of second slide with the droplet, and blood has been dispersed sidewise. This is the pushing of the blood, push forwardly at the angle of 45 degrees and 45-degree angle. And this is the final vision of the smear, which is tongue separate. Okay, so dry smear is now ready. So we have to stain it. Let us stain it. Step 2, in the staining process, we require two types of stain, 100% and 50%. But we will make it stepwise. First step, the stain will be covered with 100% stain. That means 100% leishman stain. The just cover, it has been written just cover. Why just? That means here is indicated two types of precaution. First precaution, that the stain should not spill out of the slide. Number 2, the slide should be covered with the petri dish so that the stain should not be dried out for the 3 minutes only. You have to wait and go for the next step here. In the next step, you have to dilute it to 50% with the distilled water. Distilled water will be poured on the same stain material at this proportion, at the double proportion you have added the stain in the first step. So how will you mix? It will be mixed by the pipette, in and out process. for 10 seconds. Then again cover with the petri dish and wait for 7 minutes. After that, you take it, wash out the stain from the drain of the stain from the slide, and then wash it. During washing, you should be careful that it will be kept vertically under the slow flowing of the tap water. Tap water, remind tap water, because tap water has different type of pH concentration. Now tap water, after the washing of the tap water, that means after the washing of the extra stain from over the smear, you should make it dry. Now, what will be the color? What will be the function have been ended? Two function you have to mention. First function, the smear, when during the smear has been staining. The second step, when smear is matured, smear is completed, staining is completed, then the material, stained material will be metallic vision, or the metallic color. And when the smear will be ended, then it will be a light violet color. The visual structure of the smear will be observed in the next slide here. Look, this is the blood, has been drawn in a conical fashion, that is tongue fashion. It is unstained. After staining, the structure, this color has been created. So it is the stain structure, stain smear that we are going to visit for observation. Okay, let us see the observation. Anatomical variations is found out here. The anatomical variations we want to know because we want to identify the peculiarities of the different cell structures. I have indicated only the 3 points: nucleus, cytoplasm, and granules. 3 characters are most vital. The colors have been created according to their chemical, cytoplasmic chemical reactions, and that can be checked under microscope in 45x. But nucleus and granules should be studied from this description. You see, the neutrophil. Neutrophil has the nucleus with 3 lobes: first, second, and third. It can be go up to 5 or to 7. The lobes are connected with chromatin threads here and this one, chromatin threads. Look for the eosin difference. The nucleus is bilobed and it is also connected by chromatin threads. Now, second point, this is the variation of the nucleus. Now, second point, the granules. Look for the neutrophil. The granules are fine. In case of eosinophil, the granules are coarse, conspicuous, and very much identifiable. Now come to the next cell. Basophil. In case of basophil, the granules are conspicuous. Nucleus is diffused or maybe too low. But in case of monocyte, it is conspicuous and it is kidney separate. Peculiarity is the cytoplasm outside this nucleus of monocyte does not have any granule. Granular, that is, it is a granular. But basophily is not that. Come to the next point. Agranular cells are also there. I have already told, that is the lymphocyte. Lymphocyte is two types, small and large. Lymphocyte cells have, small lymphocyte have the nucleus, round sepate, leaving a small thin space around it, cytoplasmic space. The cell shape is small in comparison to large lymphocytes. In case of large lymphocyte, the nucleus is larger, but it is eccentric in position and more or less crescent sepate, leaving more space of cytoplasm. But similar character in both cases. Cytoplasm does not have any granules. That means both are a granular. So, now come to the another cell. That is erythrocyte and platelets. The same structure is there, same character is there. That means the nucleus in case of RBC, nucleus in case of platelets are absent. And also, any kinds of granules are not found. The similarity is there, nucleus and presence of granules. That is negative. But the structure of the body shape, that means the body shape is different. In case of erythrocyte, it is round, or it can be biconcave. But in case of platelets, it is irregular in shape and present in clusters. Okay, these all the characters of nucleus and the granules have been told only to identify conspicuously when you determine or identify the cells from the field. In the field, you will see the first at 10x objective. At 10x objective, you will see the total vision. But when you focus it at 45x vision, then you will see the objective, the WBC are surrounded by many RBC cells, non-nucleated RBC cells. If you consider the different types of stainability, its color of this nucleus, of the granules, then you will find each cell by under high magnification. Here, a presumptive view have been created for your visual expression, visual identification. So, identification and the process of smearing and their color reactions I have told to you. These all structures, remember, these all structures of the cells is quite different in case of invertebrates, and that characters will be discussed in our next episode of lecture. And if you like my video and if you get some benefited from here, then share it to other students. Okay, thank you. See you in the next lecture.